You lot, welcome to my first ever YouTube video. Actually, that's a lie. This isn't my first ever YouTube video. Back in year 5, I made a video, and if you want a good laugh, search up Sam Plays space random games, and it should come up. It's quite funny. But anyway, today, what I want to chat about is this article I found about the microbiome, and in particular, it's about a specific phyla of bacteria that are within our microbiome and how they can be manipulated to potentially alleviate certain diseases like type 2 diabetes and inflammatory bowel syndrome. So I want to give you a bit of background information that you'll probably need to maybe understand the rest of this video when we get into the good stuff. And that background information will be what the microbiome actually is, and also a brief description of taxonomy. So, first of all, what is the microbiome? So, the microbiome constitutes all of the living microbes that live within us. They live in our intestines, in our colon, in our mouth, in our nose, they're everywhere. And these aren't the bad kind of microbes that you're used to that cause disease. They live symbiotically with us, within us. So we eat food, we feed them. The different kinds of microbe have different energy sources, and so as a result they make stuff. So they take this food and they turn it into stuff, and some of that stuff is useful to us. Lots of it, in fact, is useful to us. A little interesting fact is that there are actually more microbial cells, just single-celled organisms, they're tiny, than there are human cells within us. Now, I think that's quite profound. But so since they're so ubiquitous in the human body, we would expect they have large impacts on the way our body functions, and as a result, health and disease. So, second, the taxonomy. What is taxonomy? So taxonomy is the way that biologists classify living organisms, extinct or extant, not extinct. And the way they do that is by how closely related they are genetically. Or sometimes you can't get the genetics of extinct animals, so they do it based off their anatomy. It's not an important detail. But so taxonomy, how does it work? You've got non-overlapping hierarchical categories that we use to classify these animals. So that's a little bit confusing, I think. So I think a better way to describe this would be through a visual aid like this here, I hope, somewhere, and I'll just describe it a little bit as well. So you've got 8 levels of taxa or categories, and each time you go down no, that's bad. I'll start again. So, you've got 8 levels of taxa. The first level are called domains, and there are 3 domains, and that encompasses all life on Earth. So you've got prokaryotes, eukaryotes, and archaea. And so the way they're classified are prokaryotes don't have a nucleus, so before nucleus, prokaryote, and eukaryotes have a nucleus, after nucleus. And then these domains are further sub-classified into 5 kingdoms. You'll have to forgive me, I can't remember all the kingdoms, but an example would be the animal kingdom. So all animals fall under eukaryote because they're multicellular, or they've got a nucleus. But you can kind of think of it as multicellular as well. That's what you learn in GCSE, which means that organisms with more than one cell. And prokaryotes tend to be single celled, so they're just one little cell roaming around. And so these eukaryotes, you've got domains, you've got kingdoms, for example, the animal kingdom, and then the animal kingdom will be further subcategorized into phyla, and then the phyla will be, I believe, I might get this wrong, further subcategorized into order, and then family, and eventually you get down to species, and in each species there's only one. Alright, let's get into some of the results of this paper. So first I'm going to go over section 2.1, which describes the differences in the firmicute composition between diseased patients and healthy individuals. So if you remember, firmicutes are a phyla of bacteria. So a phyla would encompass a large number of species. And in this article, they describe the differences, sometimes based off the species, but usually based off the genre, which is the one kind of above species. So a genre of organisms would contain like a few species. So for example, Homo sapiens. So that's us. We belong to the genre of Homo, and we are the species sapiens. And you've got Homo erectus, Homo denisovans, and so on. So let's get into it. They found that there was a decreased abundance of Faecalibacterium in patients with type 2 diabetes, non-alcoholic fatty liver disease, and inflammatory bowel syndrome, compared with healthy individuals. Another thing they found was that patients with Alzheimer's had an increase in the genre of bacterium known as Streptococcus and Enterococcus, and they had a decrease in the abundance of genres of bacteria, namely Faecalibacterium bloatia, something like that. Can't say that. Can't speak Latin. And Ruminococcus. Yeah, the decrease in them three. But that being said, you can say that all you like, but what does that mean? That's what we want to find out. That's what we don't really know. And I'll talk more about that later on in the video when I get into the discussion and the conclusions of this review article. One thing I should mention before I move on to the next section is that it's still pretty unclear whether the change in firmicute composition, or more broadly microbiome composition, causes the disease, or the disease causes the change in the microbiome composition. Now let's get on to section 2.3, where they discuss how dietary fibres could be used to alleviate disease, because dietary fibres are actually the energy source of firmicutes. So firmicutes eat it. So if you eat more dietary fibres, logically you're going to make more firmicutes, they're going to grow. So they talk about one study that found pomelo fruits actually alleviated the symptoms of type 2 diabetes because they're high in dietary fibre, and this dietary fibre increased the abundance of firmicutes. They also outlined a paper that found a correlation between an increase in inulin intake, which is just a dietary fibre, and the alleviation of postpartum anxiety induced by prenatal obesity-induced depression. And the mechanism by which this dietary fibre does this is by increasing the abundance of lactobacillus, which is another genre of firmicutes. So the existing microbiota of a patient could affect the effectiveness of dietary fibre intervention. So more research is needed to be done in order to determine how we can treat patients through dietary fibre based off their specific conditions. And the reason this is potentially quite important is because at the minute we've got probiotics, which are supposed to influence the proliferation of beneficial bacteria in our gut. However, the safety and effectiveness of them is yet to be determined, and there's a lot of research to be done. So if we can figure out a way to use a change in diet, i.e. an increase in dietary fibre, then that would be incredibly convenient and cost effective. Okay, let's get into the conclusions and the future prospects that this paper outlines. So what do we know and what we don't know? So what we do know is that diet is clearly incredibly important for human health because of the massive impact it has on the composition of our microbiome. And this paper outlines how dietary fibre could be a very key part of dietary fibre in our diets could be a very key part of maintaining gut health, because these bacteria known as firmicutes have been proven to modulate cytokine production, and cytokines are molecules where if they're not modulated they can cause excessive inflammation and cause diseases like autoimmune disorders. And also they've been proven to help with leaky gut, so they modulate the permeability of our gut. And you don't want a leaky gut because you'll have all sorts of bad stuff going out into your bloodstream that you don't want. So what are the future prospects? Although we know these couple of things, we haven't got a full picture. All of the species haven't been studied yet, so we need to do that. We need to isolate specific species and then investigate how they behave, what kind of stuff they produce, and how they interact with disease conditions and the body in general. I hope that was interesting. If it was, I'd appreciate it if you left a comment, you liked the video and you subscribed. And with that, I'm going to leave you. Peace out.